Okay, I hope you did okay with the exercise. The second part of this session is looking at genes and transcripts. So this is how you'll have seen genes in the views that you were looking at in Ensembl. And there's a lot that you can tell about the gene just from this drawing. So the first thing you can tell is the intron-exon structure. So this filled in block is a coding exon. And joining the exons together are the introns, which are shown as these peaks. The introns are actually one of the many indications of strand that you can see in Ensembl. So you'll see that this particular transcript has its gene has all its introns in this kind of mountain style. They're also this gene is also shown above the contig, above the blue bar. And it has this arrow, and all of these things indicate to me that it's on the forward strand. If I were looking at a negative strand gene, it would be below the contig. The introns would actually be valley-shaped rather than mountains, and these arrows would be pointing in the opposite direction. We also have non-coding exons, which are shown as empty boxes. So here at the end, we have the non-coding exon, the untranslated region, which is empty. The colours are also meaningful as well. So this is BRCA2, which is a coding gene. So it has some coding transcripts, and it also has some non-coding transcripts, which are shown in blue. The protein coding transcripts are shown either in red or gold. And gold are special. They're what we call merged transcripts, which means that we identified them by two different methods of annotation. And I'll talk about those methods in a moment. And you'll notice also that all the transcripts are called BRCA2 and then are followed by a 3-digit number. So the golden annotation transcripts indicate identical annotation between two different methods, what we call the Ensembl automatic annotation and the Havana manual annotation. And we have these for human, mouse, zebrafish and rat. We do automatic annotation for every single species in the database, whereas the manual annotation is done only for this smaller subset. The automatic annotation uses a pipeline, which is genome-wide. And basically what you do is you get the sequence of the genome, you get the sequence of all the genes, and you use sequence matching to plot them all on. It's a lot more complicated than just blasting it. There's a lot of complicated things that analyse things like splice junctions, etc. But essentially it comes down to matching up sequences. All of the sequences we use are real biological experimental data. One of the misconceptions people often think when you say automatic gene annotation, it means you're just looking for open reading frames in the genome. This is not the way that we do automatic gene annotation. We have to have real biological evidence. And the biological evidence we use, so we use the INSDC, the International Nucleotide Sequence Database Consortium, which is made up of the ENA, GenBank and DDBJ. These are places where anybody can submit their sequences. And we use things like cDNAs, express sequence tags, and increasingly RNA-seq data pulled from these resources. And we also use UniProt for protein sequences. And we use both the SwissProt manually curated proteins and the Tremble unreviewed translations. If you're looking at a species which doesn't have a huge amount of species-specific data in these databases, then we will use homology. So we will predict genes in, for example, chimp by mapping cDNAs and proteins from human onto the chimp genome. Manual gene annotation is very, very different. This is done on a case-by-case basis by a real person. And the people who do this we call our Havana group. They use data from a number of different sources. So they use databases, they use papers, and they're really good at kind of weird, fiddly things that are not the norm. So things like splice sites, like untranslated regions. They put a lot of effort into kind of keeping up to date with the developments in the field and understanding what's going on so that they can annotate these as accurately as possible. there is a difference, the differences between manual and automatic annotation. There are many differences. Firstly, manual annotation is very much more comprehensive. They are aiming to get everything, whereas the automatic annotation is a lot more conservative. You tend to be a bit more cautious about what you can and can't annotate. This means you get a lot more transcripts per gene, and especially a lot more non-coding transcripts, and you get a lot more genes overall. This also means that there may be less evidence supporting them, and this is because you can evaluate each piece of evidence on an individual basis by the quality of that evidence, whereas when you're doing automatic annotation, you just have to set thresholds and make them, that's how it is. This is our line. Anything below the line we don't include. Whereas when you're evaluating things in a value-based way, you can say actually it's below the line, but it's still good enough. The manual annotation can also be a lot more accurate for things that are very difficult to annotate. So things like untranslated regions, splice sites, single exon transcripts are surprisingly difficult to annotate. And then things like the immunoglobulins, which of course are not complete genes, but bits of genes which are then that undergo somatic recombination. And these are very different to annotating many of the other genes. GENCODE is a gene set which we use in Ensembl, and it's actually made in-house. It's quite confusing that we have these different naming, but it's made up of the automatically annotated genes, the manually annotated genes, and the merged genes. It's the default gene set used by numerous projects such as ENCODE, 1000 Genomes, Nomad, GTEx, and various other major projects. And this is the gene set for human and mouse. CCDS is another kind of agreement on what is high quality. So this is the consensus coding DNA sequence set, which is an agreement between us at Ensembl and RefSeq. So wherever our transcripts match up, the coding regions of our transcripts match up to RefSeq. This then gets verified by our colleagues at UCSC, and things get a CCDS attached for them. And this is available for human and mouse. So if you're looking for the higher quality transcripts, the GENCODE transcripts represent everything that's annotated by either Ensembl or Havana or the overlap. And RefSeq CCDS represents the overlap between that and RefSeq, but only for the coding regions. We also, if you do work with rodents, we do have 3 rodents where we've imported annotation from trusted sources. The only way we know that these are high quality is because we've put them through QC, and we trust the people, and we've worked with the people that produce them, and we know that they've produced high quality data. When you start to look at transcripts, you'll see that some of them have quite a lot of some genes have quite a lot of transcripts, and it's sometimes necessary to kind of filter. One of the ways that you can filter is using GENCODE basic, and this represents only the complete transcript where a gene has complete transcript. So we will use fragments. If there's a fragment of a protein, we will plot that onto the genome, and then that may be labelled as a 5' incomplete or a 3' incomplete, or both. If you want to exclude those, if you only want the things that are finished, then you can use GENCODE basic. We have something called transcript support level, which is a score between 1 and 5 for quality, where 1 is the best. And this was produced in collaboration with colleagues at UCSC. And there's also a group in Spain who produced the APRIS principal isoform. And this is the major isoform. They combine together protein structural information, functionally important residues, and evidence from cross-species alignment to determine which they believe is the major isoform. And these are available all for human and mouse, and some of them for rat and zebrafish. If you combine this with CCDS and golden transcripts, very often you can get down to only 1, maybe 2 transcripts. We also have stable IDs in Ensembl. Most of the databases you ever come across will have some form of stable IDs. If you use RefSeq, you'll find the NMs and the NPs. In Ensembl, we have ENS, indicating Ensembl. and then a letter telling you what it is, G for gene, T for transcript, P for peptide, E for exon, and then a unique 11-digit number. If you are working with something that's not human, you'll find that there's an extra 3 letters in there, so there'll be MUS for mouse, DAR for zebrafish, and there is a list at this URL here that you can go to and find the 3-letter code for your favourite species. We also use Gene Ontology for gene function in Ensembl. And Gene Ontology, I'm not sure if you'll have come across it in this course already, but it is incredibly useful because it's a method of describing what a gene does, but it's a method that a computer can deal with. And the reason for using Gene Ontology rather than sort of free text and prose is that prose is very readable for humans, but less so for computers. So one thing that we do in language is we use multiple terms for the same thing. We use lots and lots of synonyms. So if I were to publish a paper that was describing the function of a gene and its involvement in innate immunity, that would be fine. Anyone can read that and see what it is. But if I wanted to use a computer to search for every single gene that was involved in non-specific immunity, then I might not find the paper that had used the phrase innate immunity, and vice versa. Similarly, you have sort of specific descriptions. So I could instead write my paper talking about the importance of my gene's role in natural killer cells, never ever mentioning innate immunity or non-specific immunity. Again, if a computer searched for innate immunity, it wouldn't find my paper, it wouldn't find my gene. So Gene Ontology is a way around this. One of the things they do is they form a controlled vocabulary. So there is a Go term, which I'm not going to read out, but it's for innate immune response, and it's got a description with it, and this is the terminology. This is the official terminology that Go says can be used to describe the function, this particular function, this innate or non-specific immunity. And what happens is the team at Go will read your paper and they'll say, that's the Go term that needs to be attached. And they'll put this Go term onto your gene in in their database, which will then get sucked up by many other databases, including Ensembl. So this is the term, they will read the paper, they will decide that this is what your term is. The other thing is that it forms a hierarchy. So this term, innate immune response, is actually a daughter term of another term, immune response. And it in itself has a load of other daughter terms attached to it. So if I were to write a paper describing how my gene was involved in natural killer cells, then the Go team would find this Go term, natural killer cell mediated immunity, and they would attach it onto that gene. And then somebody else were later to come along and want to find all the genes involved in innate immune response, and they did a search for this Go term. They wouldn't just find the genes that had this Go term attached to it, but they would find my gene as well with this Go term and all the other genes with all these other Go terms attached to them. So they are very, very useful ways to describe in a very computational way what genes do. If you're interested in finding out more about how gene annotation occurs in Ensembl, we do have a paper that you can look up. So I'm going to do a demo looking at an Ensembl gene. The gene is called ESPN, which is involved in actin bundling, and it's known that mutations in this gene cause deafness. So we're going to find out information about it and its transcripts. After the second demo, then you can have a go at task 2 on your worksheets.